about 4 years ago, actually. I believe even before I formally joined the company, we filed a patent on that. And that reminds me something that I need to ask you guys. So the cells were injected in the muscle, but the patient started getting better erectile function. So that reminds me, there was a study recently published, I forgot the journal, but where they were doing the myocardial ischemia model, giving a heart attack to the rat, but implanting the bone marrow cells intramuscularly in the thigh muscle, not into the heart. And they were seeing almost the same effects as if you were injecting the cells directly into the infarct-related artery. The study believing that it's all paracrine, not even paracrine, endocrine effect, that the cells are just producing these different factors that act systemically. What do you guys think? Is that, can a lot of these things just be endocrine, so we can just inject supernatants? We don't even need cells? Is that a possibility?
It's a good question, and there's been a lot of animal work related to it. But what ends up happening is, unless you have a very specific process, such as ischemic heart disease, and you get the cells cultured in the ischemic medium, it makes it very difficult for just the paracrine factors alone to give you the end response that you get with cells. That's the great thing about cells, is when you put them into the diseased environment, they sense the local environment and then determine what paracrine factors are going to be released, as opposed to predetermining it in vitro. You just don't see the same response. Even though we see a subtle response in animals, we don't see as dramatic as a response as when we directly inject cells or the cells are in the diseased environment.
Now, that's a very, very... That's a very, very interesting point, and that makes me revert back to what we were talking about previously, what Lee brought up is the safety considerations. There was a study in which, I believe you guys have probably seen this 3 years ago, in which they give to the mouse Helicobacter infection, and they inject in the mouse GFP, they make the mouse chimeric, so that the mouse has blood, green blood, basically, and they give it the Helicobacter into the stomach. The mouse then develops a gastric cancer. The gastric cancer, because of the constant inflammation of the Helicobacter, the gastric cancer was all donor bone marrow derived. So the bone marrow cell is differentiated into gastric tissue, and then the gastric tissue became cancerous. Has there been any other examples of stem cells turning cancerous?
I think your stem cell analogy here goes the same as with handguns. Guns don't kill people, it's people kill people. The cells don't actually cause the cancer, but they'll really make it a great environment that if you're already predisposed or have early cancers, it will make it go very rampant and it will kill the patient. So the cells are not bad. It's the use of the cells in the wrong environment for the wrong reasons can become very bad.
Because exactly. But see, this is very, very interesting, because we also published a paper recently which we were shocked at the data, which is our cells, endometrial regenerative cells, we're using them to stimulate angiogenesis. So as part of our studies for the FDA, we, number one, wanted to make sure the cells themselves don't become cancerous, and they didn't. And then we wanted to give the cells to pre-malignant lesions. Basically, we were using a mouse model where you irradiate the skin of the mouse, and then after 2 months, the mouse develops a type of melanoma. When you give the ERCs in this model, it seemed like there was an inhibition in the development of the spontaneous melanoma, of the UV-induced melanoma. So that really shocked us. It was a small study. So we actually made that study bigger and looked at a glioma model, the C6 glioma model. We injected the glioma cells, injected ERCs. The ERCs not only inhibited the glioma growth, but actually inhibited the angiogenesis. And you have to read the discussion to the paper because basically in the discussion we said we really don't have an explanation for this between the lines. Do you guys have any ideas? Could stem cells in certain situations actually be inhibiting cancer? I mean, Lee, you talked yesterday about anti-inflammatory aspects of stem cells. People talk about cancer as being the wound that never heals.
It is. But so Tom, it's a good question because what you see is two effects. So as we see in the mesenchymal cells, the anti-inflammatory response, you see in the dendritic and NK cells, you can actually make cancer vaccines using autologous cells and the patient's own cancer, and you use that as a priming system. And using your own stem cells, there's still components within that population that can go down the pathway of a vaccine-like process. So you have different components. You have the hematopoietic, you get the angiogenic component of that. You get the stromal cell, which has the anti-inflammatory. But you also have the NK cell, dendritic cell population can all be derived from stem cells that can have the true anti-tumor or vaccine-like effects like you see with the Dendrion product. So it is possible to have a vaccine-like therapy. So it's true that stem cells in a bad environment that are just the pro-angiogenic and also even some of the stromals can actually enhance a cancer response, whereas if you can steer them down the pathway of more of a dendritic NK cell, then they are actually going to kill your cancer or actually be anti-cancer. So that's the great thing about stem cells is depending on the environment and the right cues and signals that they get can really drive which pathway they belong to and what your end result is.
That's
well, guys,
right. That's the thing. I think it can go both ways.
Lee, hold on 1 quick second. On the chat there, this is under your screens, I guess. For some reason, it was disabled. So I want to say to the people watching, I missed your questions. When the people are watching, if you can just type in questions and we can see them. Sorry, Lee, go on.
I'm sorry. Do you want to answer some questions now, Tom?
Well, I'm not seeing any questions yet. Apparently, it was disabled.
Okay.
But
okay.
Look, somebody
said.
No, I was. Somebody said, "Wow!" Somebody said, "Wow, this is live," and somebody else said, "Hello, Tom. Hi." Okay, sorry. Go on.
So I think that's both the beauty and the potential caution that we have with these therapies is A, there's a lot we don't know, and B, these things can do a lot of different things. The great irony is that someone who's really healthy, who's a runner, for instance, who has a great fast metabolism and has a highly active and mobilized circulating stem cell population might be doing themselves harm in terms of the growth of their own cancer.
Oh, look, guys. Sorry, Lee. Does he work with Tom? No, Lee works at at Cell Therapy Group. Cell Therapy Group. Now, okay, somebody's saying here about the human cord MSCs in the breast cancer model. So this was a study that was a very interesting study where what they do, this is mismatched. This is human cells going into a rat. And the rat has an aggressive breast cancer. I believe it was the 4T1 strain. And the cells actually completely cured the rat breast cancer model. So that's what I know about the study. Maybe the person who asked about it, maybe they have some other things that they want us to say. Okay. I guess nobody else has any questions up there. Okay. Very bizarre way to do this. So, the one last point with the cancer, from our work, what we're doing is we're working with Boris Miniev at UCSD. He's transfecting ERCs with TRAIL, the tumor necrosis factor analog TRAIL. Basically, it's like a poisonous payload. Transfecting the ERCs, injecting them into the animals so that the ERC is home to the tumor. Once they get to the tumor, he actually has a visualization system with microbubbles which he can activate. So anyway, that's the conversation about cancer. Okay, one last thing, I guess, before before we'll let you guys go, since we have you, Amit, here, and you're the father of this field with respect to cardiology, how far are we right now? I mean, to getting one, as an industry, how far are we to getting something FDA approved or a major regulatory body approved? Like you said, it's been at least 10 years since the work started with bone marrow in the heart and Stephanie Dimler and all those groups. How far are we now, would you say?
Realistically, for a cardiac disease, we're still probably close to 5 years away before we have a fully approved product. There have been a number of completed phase 2 trials in the U.S., but for many reasons, we have not initiated any true phase 3 trials. And we're hoping by next summer, there'll be at least 2, if not 3, phase 3 true cardiac trials in the U.S. Whereas vascular disease for critical limb ischemia, the trial started much later, but the results were so dramatic that independent of which group and what part of the world, people are seeing up to a 50% reduction in amputations at 6 months. So as opposed to cardiac, which had great promise, but the problem was some people were treating angina, some people were treating heart failure, some people were treating acute MI. So the problem was for the general patients and population, everyone was looking at ejection fraction. So they were just looking at the endpoints in the wrong patient population. So it gave a very mixed message, which I think really slowed down the field. Whereas critical limb ischemia, it was very black and white. At the end of the trial, you either had a leg or you didn't have a leg. And the results were so dramatic that even in the worst trials, you were still seeing a 30% reduction in amputation. In the best trials, it was 50%. So now you have at least 3 groups. You have, the Harvest guys, you have Astrum, and you have Biomed, all basically running neck and neck in phase 3 trials in the U.S. for critical limb ischemia. So it's very possible that in the next 18 to 24 months, we could have not only completed phase 3 trials, but the potential for a realistic approved product that has a very dramatic effect. And that's probably what will really catapult a lot of the other cardiovascular diseases to move probably at a quicker rate, but also just more cleaner trials, because everyone has the same questions. Where do the cells go? What do they become? What are we really changing? Whereas in limb ischemia, it's very simple. You either have a leg or you don't have a leg. The wounds got better or they didn't get better. So it makes it very tangible, not only for the patients in terms of their quality of life and functionality, but also at the other end for the scientists, the functional and microscopic things are very realistic in terms of angiogenesis, in terms of improving circulation, very objective parameters. So that's why critical limb ischemia really is the best, predictor or force in cell-based therapies that has the potential being a fully approved product here in the U.S. in a very short amount of time. Cardiac, still 3 to 5 years, even though there's incredible results. All of us have treated heart failure patients who have had these dramatic responses, but it's just adding to the reproducibility of the therapy and really trying to get a cleaner population, just because when you try to treat patients who have multiple different causes of heart failure, it's hard to get clean endpoints that are reproducible to get an approved product. So I think the field is still progressing, and I'd say in this last year or two is what's really accelerated the field. The first 5 to 8 years, there was a lot of great stuff happening outside of the U.S. As patients always ask us about what they should do, I always say there's so many things going on here in the U.S. We can help majority of the patients with cardiovascular disease now.